In the second part of photosynthesis in higher plants, let us learn what are the raw materials that which is used in the photosynthesis. So what will be the raw materials that which is used? So leaf to stroma, how it is taking place, we will learn here in this part. So as I have told you, carbon dioxide, where do we get carbon dioxide? From the atmosphere. Where do we get water molecules? We do get it from the soil. So we know that carbon dioxide is received from the atmosphere and water molecules we do obtain it from the soil. Okay, so here in this above equation, what is important is that why 12 molecules of H2O are used here. So here in this above equation, the important question is that why 12 molecules of H2O are being used. Why 12 molecules of H2O are being used? So in the Calvin cycle, we will learn later in the third portion, we will learn what is Calvin cycle, how it is being formed, all those things we will be learning. So if we wanted to make one molecule of glucose, then we have to 6 times we have to run the Calvin cycle. Then only we will be getting one molecule of glucose. So to run one Calvin cycle, we need 2 NADPH. That means what is NADPH? Nicotinamide adenine dinucleophosphate. That's an enzyme used here. So 2H plus is also required. Where this H plus comes from? We all know that when we break the 2 molecules of H2O, we will get 2H plus. So we can come to a conclusion that if 2 NADPH is used, then 2 molecules of H2O should be broken. So 2 molecules of NADPH को चाहिए तो हम लोग को 2 molecules का water molecules को break करना ही होगा, split करना ही होगा. So if we need 12 molecules of NADPH, then of course we have to break 12 molecules of H2O. So that's what the 12 molecules of H2O is being used in the above equation. So next here is the light. Light, photosynthetically active radiations. PAR means photosynthetically active radiations. So generally this light comes from electromagnetic radiation, that we know that sun, from the sun. Ultimate source is sun. Okay, so here the absorption rate will be 400 to 700 nanometer. So what are the two functions of the light here? Photolysis of water molecules, at the same time excitation of chlorophyll. So here photolysis of water molecules. So photolysis, due to photolysis, what is happening here? So due to photolysis, protons, electrons and oxygen is being released to the atmosphere. In the photolysis, breaking of light, lysis means breaking, photo means light. So here in the photolysis, protons, electrons and oxygen will be released to the atmosphere. And what these protons and electrons will be making? They will be making the ATP and energy and NADPH2. So what is this ATP and NADPH2? These are the assimilatory power here. Then these are the assimilatory power. That is very, very important that you have to remember. So in the photolysis of H2O, protons, electrons and oxygen. Oxygen is released to the atmosphere. Protons and electrons will be forming ATP, energy, that we have to remember. ATP is adenosine triphosphate. ATP and NADPH2 is, they are the assimilatory power that we are using here. And next we have alignment of chloroplast. So what is the chloroplast? What is the alignment of chloroplast? We all know that the cells में तो mesophyll in the leaves, when we have taken in the practical, we have done it. So when we are taking the section of the leaf, we do see the mesophyll cells, the palisade cells, the spongy parenchyma, the chlorophyll, how the pigments are being present in the leaf, that all we have seen in the practical. So in the laboratory we have seen it. So how the chloroplast is being arranged in the mesophyll cells, that is very, very important. So now let us learn it. So here we have the alignment of chloroplast. You can see here the alignment of chloroplast. So when the high intensity light is given, then this will be, when the high light intensity is given, it will be parallel. Okay, the alignment of the chloroplast will be parallel. When the low light, it will be perpendicular. You can see here it is perpendicular. And when the moderate light is given, it is being randomly put. That means it is बिखरे हुए पाएंगे. So we will be, we can see that here spreading all over. So when the moderate light is given, उसको हम बिखरे हुए पाएंगे. That means it is randomly or separately. These molecules will be, the chloroplast will be in the randomly put. Okay, so these three things you have to remember. When the high light intensity, the chloroplast will be arranged in the parallel level, and when the low light intensity, then it will be arranged in the perpendicular level. Then in the moderate light it is random. Okay, so these three things you have to remember: when the high light intensity, and in the low light intensity, and in the moderate high light intensity, how the chloroplast is being arranged. That is very, very important. So next we will learn about the photosynthetic pigments. Where these pigments are present? These pigments are present in the thylakoids. Okay, location is of thylakoids. And another thing that we have to learn here is that the deficiency of minerals are known as chlorosis, and the deficiency of light is known as etiolation. The deficiency of minerals are known as chlorosis, and the deficiency of light is known as etiolation. So these two things are important. Now let us learn about the pigments. So here now we have the pigments, the important pigments: chlorophyll A, chlorophyll B, xanthophylls, carotenoids we have. So the color variation also is there. What are the functions? Let us learn of the chlorophyll A. So chlorophyll A, we can call it as chief pigment, or we can call it as universal pigment. It is involved in the photosynthesis process, and another function is that it is essential. Okay, it is essential and a reaction center. It is taking place involved in the photosynthesis process. It is essential. Okay, that we can consider it as the universal photosynthetic pigment. Okay, chlorophyll A is used everywhere. Okay, हर एक photosynthetic organism में क्या है? Chlorophyll A है. Chlorophyll A is present. यूबैक्टीरिया को छोड़कर हर एक फोटोसिंथेटिक ऑर्गेनिज्म में क्या है? क्लोरोफिल ए है. सो दैट्स व्हाट इट इज कंसीडरिंग एज द चीफ पिगमेंट. And next we have chlorophyll B here. Chlorophyll B, xanthophylls and carotenoids are considered as accessory pigments. Accessory pigments, why? So now let us learn. Because it is not directly involved in photosynthesis. So the chlorophyll B, xanthophylls, carotenoids are considered as, their functions considered as accessory pigments. Why? Because it is not directly involved in the photosynthesis. Then how we will be having a doubt? Then how it is helping? How it is helping? These are absorbing the light in different wavelength. So we will see in the previous slide. Then we will go to the previous slide. So we can see that how it is being absorbing in the various wavelength. Then it will be passing through the reaction center. Light means photon. So it will be passing through the reaction center. And another thing is that why it is considered as accessory pigment is that they will be protecting chlorophyll from the photo-oxidation. So what is photo-oxidation? So when the chlorophyll is distracted by the high light intensity, then we call it as photo-oxidation. So it will be protecting the chlorophyll from the photo-oxidation. That's what it is considered as accessory pigments. Then it enhances the efficiency of the photosynthesis. Another important point is that it enhances the efficiency of the photosynthesis. So this is what these carotenoids, xanthophylls and chlorophyll B is considered as accessory pigments in function. Next, moving forward to the spectrum. So we have action spectrum and absorption spectrum. What is the meaning of action spectrum and absorption spectrum? Absorption spectrum means absorbing the light in various wavelengths. Action spectrum means in which rate any of the things are being absorbed. किसी भी चीज़ को किसी भी rate में absorb करते हैं तो उसी को action rate बोलते हैं. So action rate and absorption rate. So here we have the absorbance of light by chloroplast pigment. So absorption of light. So here we can see the maximum absorption with geo. We have the blue is having high intensity, the red is also having high intensity, green is having little less. So that is in carotenoids, chlorophyll A, chlorophyll B. So the high intensity light that which is taking maximum absorption is taking place in blue and red. We can see here in this image. Moving forward, that is the rate of photosynthesis. So rate of photosynthesis also we can see that the absorption rate, how the absorption rate is. So the maximum is taking place in the red and blue. So in the 450 nanometer. Next we have here the rate of photosynthesis and absorption. So these, they don't overlap. You can see here the rate of photosynthesis and the absorption. So 450 nanometer to 700. That is the range given here. So in our first class, as I have told you, P700 and P680. So I hope now you might have understood. So the absorbance of the light in the nanometers, that is P680 and P700. So rate of photosynthesis and absorption. So that does not overlap with each other. So here the main thing that which is being asked is that what is the most abundant pigment? That is chlorophyll A. Okay, the most abundant pigment is chlorophyll A. And next question he has most probably asked is that where the maximum absorption is taking place? In the blue. So that is within 450 nanometer. So in blue light, maximum absorption will be taking place and in 450 nanometer. And the next question is that does it overlap, the rate of absorption and the rate of photosynthesis, does it overlap? So that does not overlap. So photosynthesis will be taking place in the blue as well as the red regions. That's the important thing that we have to remember here. So next important question is this: where the photosynthesis is taking place? Where the photosynthesis is taking place? So we have already done the practical, the section of the leaf we have taken. So we have understood how the chloroplast is being present, how the mesophyll cells are being arranged, how the palisade cells seems to be, how the spongy cells, how the cells are being arranged in the leaf. We have clearly seen through the microscope. So those who have attended the practical, they have seen. So here I have zoomed one leaf. So here you can see here the leaf. When we take the section, so sectional view, we can see here the mesophyll cells, the palisade cells, the upper epidermis, the vascular bundle, lower epidermis, substromal cavity. We can see here the xylem, phloem arrangements, the palisade cells, the spongy parenchyma. All these things we can see. We have seen also when we have done the experiment. When we have taken the section of the leaf, we have seen how the chloroplasts are being arranged, how the vascular bundles are being arranged. Okay, so moving forward. So here we have the sectional view where the photosynthesis is taking place. The photosynthesis is taking place in the stroma lamellae. Okay, we have here the stroma, the sectional view of chloroplast here. So stroma lamellae in the thylakoid region, this is thylakoid region, and the connection between the thylakoids and stroma lamellae, and this is the stroma region. So in the lumen. So we have here the palisade cells. We have taken a sectional view of palisade cells also. So how the chloroplasts are being arranged, the nucleus are being arranged, that also we can see here. And here we have zoomed the granum, a stack of thylakoids. As I have told you, the granum has been arranged like the coins. So a stack of thylakoids are being present here, and how the inner membrane of the stroma is being arranged, how the hydrogen ions, how the ATP is generated, how the energy is released, all these things are here. And next, moving forward to the light reaction as well as the dark reaction. So what is light reaction, what is dark reaction, where it is taking place? We have stroma here, we have granum here, and we have chloroplast here. So light, it does not mean that light reaction is taking place in the light and dark reaction is taking place in the dark. No. Light reaction, of course it is taking place in the light, but with the product of this light reaction, dark reaction does not depends upon the light, but with the product of the light reaction, this will be utilizing energy and preparing food. So that's all about the raw materials of the photosynthesis, and in the next class we will learn the photosynthesis, mechanism of photosynthesis. So thank you for listening. Thank you. May God bless you all.